Morning everyone, this is GS Academy, Ramburam, Chennai, CSIR NET, Life Science Unit 5. This class is about developmental biology, the role of differential gene expression. Let us see what is morphogen. Morphogen gradients provide positional information during the development. Cells often need to know where they are with respect to the body as a whole, as in the case of the whorls in the flowers, just described in the last class. This spatial sense is called the positional information. So, the positional information usually comes in the form of a signal that is called as morphogen. So, what is morphogen? Morphogen is a signal that usually comes from the positional information. So such a signal is called as morphogen. You can see the plant. This is the plant which is called as Arabidopsis thaliana. So these morphogens are very important to send the signals to different areas to produce different whorls in the flowers. Okay. So the auxin gradient mechanism which generate the distal root in the Arabidopsis thaliana is different from the earlier studied mechanism. So three distinct mechanisms of morphogen gradient formation we are going to see now. The first step is source decay mechanism. What is this source decay mechanism in the sense? The overall morphogen production is localized at the source and decay. This mechanism is called as source decay mechanism. Next one is unidirectional transport mechanism. So what is this mechanism in the sense? The morphogen is directly transported to the direction of to the dead end. Wherever it is necessary, to the dead end, this will be transported. The morphogens will be transported. So that is called as unidirectional transport mechanism. Either it is on the tip or at the root or at the lateral. So that is called as unidirectional. It is in only one direction. Okay. And reflex loop mechanism in the sense, it is the combination of a downward or upward or the lateral flex. So there are three mechanisms: source decay mechanism, unidirectional transport mechanism, and the reflex loop mechanism. Next, if you see the fact that these morphogens specify these axes was established by the result of experiments in which the cytoplasm was transferred from one egg to another. So the females that are homologous for a particular mutation of the maternal effect gene, the gene is called as bicoid, that produce larvae with no head and no thorax. You can see here. And however, if the eggs of these females are inoculated at the anterior end with the cytoplasm, from the anterior region of the wild type egg, the treated eggs develop into normal larvae. You can see this is the bicoid mRNA and this is bicoid protein, and the transcriptional activations forms these mRNA. This is hummock mRNA and the caudal protein, and this is orthodentical mRNA. So after transcription, these three types of protein as well as the mRNA are formed. If you see this one, this is segmentation and homeostatic. Before going to see that, we can see the role of differential gene expression establishing body segmentation of Drosophila melanogaster. Okay, what you are seeing here is the cell. This is a single cell, egg cell, and this is the nucleus, and these are the nerve cells, and the outer covering is follicle cell, and this egg is a single nucleated cell. Okay, and after fertilization, What you are seeing, this is after fertilization, and the embryonic development, the segments are slowly formed, and this is the larva. This is the embryo, the body segments you can see, and this is the larva, it can move. Okay, and if you come to see this one, the insect, that is the Drosophila melanogaster, develop a highly modular body composed of different types of segments, what you are seeing here. And the Drosophila egg as well as the larvae are characterized by unevenly distributed cytoplasmic determinants. See, if you see this this cytoplasm, in the sense there are so many cytoplasmic determinants. These cytoplasmic determinants are unevenly distributed in the segments, and these molecules, which include mRNA as well as the protein, and they are the products of specific maternal effect genes. The maternal effect genes that are comes from their mother ovarian cells, and these nurture the developing egg. Those proteins nurture the developing eggs, and the maternal effect genes exert their effect on the embryo regardless of the genotype of the father. And the product establishes the dorsal ventral, that is back belly, and anterior posterior, that is head to tails, access the embryo. And if you are seeing here, this is the egg with a single nucleus. So the cell won't divide, the cytoplasm won't divide. The nucleus divided into many nucleus. So this is a cell with multi-nucleus. So what we are seeing here is the larval segments are determined by the proteins encoded by the segmentation genes. All these genes are expressed when they are about 6,000 nuclei in the embryo. This is the embryo, in the sense, if it comes 6,000, the nucleus number is 6,000, then only the genes are expressed. Okay, which gene? In the sense, segmentation genes are expressed when this cell reaches 6,000 nuclei. Okay, these nuclei are looked the same, but in term of gene expression they are differ. Genes are different, but they all looks alike, but the genes present inside the nucleus are differ. Okay, the product of the maternal effect genes set the segmentation genes in motion, and three classes of segmentation genes are enact one after other to regulate the finer and finer details of the segmentation pattern. So what are the genes, three genes we are going to see now. The first gene is called as gap genes. Second one is pair rule genes. Third one is segment polarity genes. What is gap gene? In the sense, these gap genes, the duty of the gap gene is organize broad areas along the anterior to posterior axis. Anterior to posterior axis, it organize the broad areas. It organize the broad areas from anterior to posterior. And if you see the pair rule genes, in the sense, they divide the embryo into units of 2 segments each. And mutation takes place in the pair rule gene. In the sense, what will happen? It results the embryo missing every other segment. So the segments will be missing if any mutation takes place in the pair rule genes. If any mutation occur in the gap gene, in the sense, the omission of several larval segments takes place. Okay, so omission of several large larval segments takes place when the gap gene got muted. And if it is pair rule gene get muted, in the sense, in embryo missing every other segment, other segments will be missed. If you take the segment polarity gene, the main function of this gene is determine the boundaries and anterior posterior organization of the segment. So they determine the boundaries as well as The anterior to posterior organization of the segment, the third one does. And if you see the last one, what it is in the sense, homeotic genes. These genes are finally, after the basic pattern of the segmentation has been established by the segmentation genes, difference between the segments are mediated by the activities of these genes. Most unfertilized eggs are the storehouse of mRNA, which are supplied by the mother and support the protein synthesis during early stages of the embryonic development. Indeed, the zygote and the early embryo do not carry out the transcription. Only after several cell divisions does transcription begins and forming the mRNAs needed for later development. Let us see what stage is known as syncytial stage. What is syncytial stage in the sense? In Drosophila, see these are the early development stages of Drosophila, and we can see these two stages are called as syncytial stages. Why it is called as syncytial in the sense? The cytokinesis does not begin in this stage. Only the nuclear division takes place and the nuclear arrangements towards the periphery of the egg, that is the zygote takes place, and there is no cytoplasmic division. Okay, so until the 13th nuclear division, the embryo is single. The multinucleated cell called the syncytium. So the stage is called as syncytial stages. And what you are seeing here is this is dorsal, this is the adult Drosophila. It lays the eggs in the sense this is a fertilized egg, and the nuclei divided into many nucleus, and the cytoplasm also consists of cytoplasmic determinants. And this is the dorsal area of the zygote, and this is the ventral area. This is anterior area of the zygote, and this is posterior area. And during the development, the nucleus move towards the peripheral area, and then the cytoplasmic division takes place. Okay, the peripheral area, you can see a lining of the nucleus and followed by the cytoplasmic division, and this is now the cellular blastoderm, and then protein gradients and the segment formation in this is the embryo of the Drosophila. Okay, at 10 hours, after 10 hours of the zygote formation, and this is the adult Drosophila. And in the early point, you can see the bicoid mRNA is translated forming. If it is from the bicoid mRNA, the bicoid protein is translated, which diffuses away from the anterior end established by a gradient. And at the posterior end, anterior end, which protein in the sense it is bicoid protein. The translation of bicoid protein from the bicoid mRNA, and the posterior end or the posterior region, the nanose protein forms a gradient in the other direction, in the sense in the posterior end region. Okay, and each nucleus in the developing embryo is exposed to a different concentration of ratio of bicoid as well as nanose protein. Okay, the two morphogens regulate the expression of the gap genes, although in different ways. The bicoid protein affects their transcription, while the nanose protein affects their translation. You have to take this, note this point. Two morphogens regulate the expression of the gap genes, although in different ways. How? In the sense, the bicoid protein affects their transcription, while the nanose protein affects their translation. Okay, translation of these gap genes. And the high concentration of bicoid protein in the anterior portion, that is, if you take the larvae, if this is the larvae in the sense, anterior portion, it is the high concentration of bicoid protein in the anterior portion of the egg turn on a gap gene called hunchback. Hunchback. While simultaneously the turning of another gap gene which is crupple. So these two are very, very important. The gap gene, if it is in higher concentration in the anterior one, then it is called as hunchback. Simultaneously turning off another gap gene that is called as crupple. So there are two gap genes. One is hunchback and another one is crupple. And there are two bizarre homeotic mutation takes place in Drosophila. What are in the sense? Antennapedia mutation. What is antennapedia mutation in the sense? The legs grow in place of antenna. This is very important. You make a note of this. Antennapedia mutation. Antennapedia in the sense in which the legs grow in the place of antenna. Instead of antenna, the legs grow at this region. So this mutation is called as antennapedia mutation. Okay, this takes place in the Drosophila. This is the head of the Drosophila. And bithorax mutation. So two mutations occur. One is antennapedia mutation and another one is bithorax mutation, in which the extra pair of wings grows in a thoracic segment. Extra pair of wings grows. Okay, so that mutation is called as bithorax mutation. And one more important point, you have to see that Edward Lewis at the California Institute of Technology found the antennapedia and bithorax mutations, and not of isolated genes but of two adjacent clusters of genes that determine the identity of the body segment. This was found by Edward Lewis. And what is homobox? What is homobox in the sense? The 180 pair of DNA sequence that is a common to bithorax as well as antennapedia gene cluster is called as homobox. So what is homobox in the sense? 180 base pair of DNA sequence that is common in the bithorax as well as the antennapedia gene cluster is called as homobox. And another one, if you see, it encodes a 60 amino acid sequence which is called as homeodomain that binds to the DNA. The homeodomain turns out of the present in another protein involved in the Drosophila pattern formation such as bicoid. So the homeodomain portion of the protein has a turn helic motif. Each type of homeodomain recognizes a specific DNA sequences which promoter of its target genes. So what is that in the sense? The bicoid homodomain, for example, let us take, it recognizes TCC, TAA, TCC-C. Okay. And gene regulation. A number of genetic components of aging have been identified using a model organism. Which organism in the sense? Budding yeast, Saccharomyces cerevisiae, and the worm C. elegans. This already we have seen in the last class, as well as the Drosophila melanogaster. So the gene regulation is best studied in these three organisms. And aging pathways. If you see the aging pathway, one of these pathways involves the gene CSIR NDA plus. These are dependent histone deacetylase. These enzymes are dependent histone deacetylase. In yeast, if you take, SIR2 is required for genomic silencing at third loci. This point is very, very important. This happens in yeast. Okay, SIR2 is required for the genomic silencing at 3 loci, and the yeast mating loci, the telomeres and the ribosomal DNA. Okay, this is required for the genomic silencing at 3 loci. Number 1 is yeast mating loci, the telomere as well as the ribosomal DNA loci. Okay, these 3 loci, this SAR2 is required. And in some species of yeast, replicative aging may be partially caused by homologous recombination between the rDNA and excision of rDNA, which repeats the result in the formation of extra chromosomal or DNA circles. Okay, you can see the extra chromosomal circular DNA here. This is uneven secretion of the circular DNA between the daughter cells, uneven distribution. If this cell divide, in the sense, this uneven distribution of the circular DNA between the daughter cells. These DNA which replicate and preferentially segregate to the mother cell during the cell division, and these are believed to result in a cellular senescence by titrating away the essential nuclear factors. The extra chromosomal circular DNA, that is ECC DNA, has been found in worms, flies as well as in human beings. So where we find this extra chromosomal circular DNA, in the sense it is found in worms, flies as well as in human beings. Aging is a gradual and functional decline of cell. Aging is a gradual and functional decline. When age grows, automatically the function will decline. So in mammal, if you take, aging occurs in multiple organs heterogeneously, that causes progressive deformation of tissue, and finally the tissue become dysfunctional. And aging is a risk factor for several diseases such as dementia, osteoporosis, cardiovascular disease as well as pulmonary fibrosis and glaucoma. And factors involved in the aging divided into 3 categories. One is primary, and next one is antagonistic, and third one is integrative. What is primary? In the sense, in this type of aging, the age associated with the damage, okay, the antagonistic, in the sense it responds towards the damage. And third one, if you take this senescence, is a type of cellular responses that limits proliferation as well as the aged or damaged cell during the normal development of cells. Thank you so much for watching this video. Share this video to your friends, those who are preparing for the CSIR NET. Thank you so much.